Hi class, Dr. Jim here. In this set of lectures, what we're going to be looking at is how the cell divides. And so you need cell division in order for the cells to grow and to divide and to make new cells for when you need growth and repair of your body. And so this is an important concept that we need to look at. Now, the last set of lectures, we looked at how the DNA prepares itself and gets ready to go from one double-stranded piece of DNA to two. And that's really important because we need our cells to replicate its DNA first before we can divide into two cells. And so these things go hand in hand. So if you remember watching those lectures last week about DNA replication, this is what we're going to be talking about today. Now we're going to be looking at how the cell divides. So we looked at how DNA replicates, and that's part of what is involved with cell division, and that's what we're going to look at. And so today's lecture, what we're going to focus in on, what is the cell cycle? The cell cycle is basically what the cell does in order to replicate and get into two new cells. Okay, we go through replication in DNA, and then it divides into two cells, and that's really the cell cycle. All right, and then when we get done with that, we're going to look and see what happens when things go wrong, and that's the cancer itself. And so those are the two topics we're going to hit today. So what are our objectives? Well, first we're going to look at what is the cell cycle. What I like to picture the cell cycle as a big clock. So think of a clock on the wall or like Flavor Flav War, and basically look at it that the cell starts at 12 o'clock. And as it goes around through the cell cycle and ends up back at 12 o'clock, what happens at the end of the cell cycle is now that you have two new cells. Okay, and that's what we're going to look at with the cell cycle. We're also going to talk about why do we even need one? Why do we need cell cycles? Why can't the cells just stay in happy suspended animation? Why do we need to make more cells? And we'll explain why we need to do that. We'll talk about what happens when everything goes right and then when everything goes wrong. And so what happens to the cells if they don't go right? This happens all the time. Sometimes the cells grow abnormally, sometimes they divide incorrectly, sometimes the DNA doesn't replicate correctly. And what happens to those cells? This happens all the time, but we don't die of cancer every day. And so what happens is we have mechanisms that actually avoid this situation. We're going to talk about that. And then finally, we're going to look at what is cancer. And so I'm going to talk about what cancer is, which is basically an abnormal growth of ugly-looking cells, and why this actually happens. Like I just said, or I just mentioned, we have all these checks and balances to keep cells from actually undergoing cancer. But sometimes what happens, our cells just miss the whole checks and balances, and they end up abnormal and start growing out of control. And that's really what cancer is all about. And so we're going to look at all three of these things today. So the first thing is, why do we even need a cell cycle? And I brought this up the last time when we talked about DNA replication. We need this in order to grow to an adult. So we all started as babies, and we got bigger, bigger, and bigger, and taller. And that in order to do that, we needed our cells to divide and get bigger and divide and bigger and divide so that we become an adult. The other thing we need this for is to heal. We cut ourselves, we scrape ourselves, we injure ourselves all the time. And if we didn't have the cell division, we'd have open wounds all over us. We'd be a mess. Okay? And in order not to be a mess and look pretty and all these things, we need to heal ourselves. And the way we heal ourselves is undergoing through a cell cycle and mitotic phase. And so we'll look at that as well. The other thing we see every day is the plants outside. The plants need to undergo mitosis in order to grow and become full-fledged plants so that we have fruits and vegetables every day to eat. And again, we're going to look at this in the lab this week, mitosis with the onion root. And you're going to see lots of mitosis going on in these cells. And in order to grow and divide, they need to undergo mitosis as well. And cell cycles are really important there too. Okay, so let's move on. And so here's the cell cycle. And like I said, picture a clock. All right. And if you picture a clock and you start at 12 midnight, you start with one cell. And it works its way all the way around to about 11 o'clock the next day. And when you get to 11 o'clock to midnight, this small little phase is called mitosis. And mitosis is where you actually go from one cell to two. And that is the mitotic phase. And we're going to talk about that in the next lecture that accompanies this. I really want to spend some time just on mitosis, so make sure you watch that video to understand the process of mitosis. I'll briefly mention it here, but go in more detail in mitosis itself. The rest of the phase is called interphase, from 12 o'clock up here all the way to 11 o'clock here. This I, or interphase, is an important part of getting the cell ready to divide. It's actually divided into three phases called the G1, the S, and the G2. And we're going to explain what those things are in just a minute. But just to kind of give you a heads up, G1 means gap 1 or growth. S is synthesis, where you actually replicate your DNA. And G2 is then you grow, continue to grow until your cell is ready to divide. Okay, and then you hit the M phase, your cell goes through mitosis, and then you end up with 2 new shiny cells out of the one that it started with. And so this is the cell cycle. All right, so let's look at the cell cycle more in more general terms, okay? Or actually more specifically for these different phases. So again, the orange represents interphase, and interphase is about 90% of the time. And if we look at the individual phases of interphase, we have the G1, and G1 is just the growth phase. This is where you start with a small cell and you continue to grow until you get ready to replicate your DNA, and that's what G1 does. It grows the cell until it's ready to replicate its DNA. However, there's this little G0 phase, which is basically where the cell stops, and it's called the suspended animation, where the cell actually enters this phase. Not all cells do it, but there are a couple cells that do in our bodies, and that is nerve cells and that is muscle cells. And once they enter this phase, they never come out of it. And so that's why they always say the adage, if you drink, you end up killing brain cells, because brain cells never come back. It's also the reason why, like, if you suffer a severe spinal cord injury, you can't repair yourself because those cells never grow back. And so that is the issue there. We have new treatments and new revolutionary treatments that are in the pipeline, looking at stem cells to try and repair those cells. But for the sake of this lecture, think of nerve cells sitting in this G0 or suspended animation phase, and that they never go past this G0 phase. Okay? But any other body cell actually goes through the G1 and then enters into the second phase, which is called the S phase, or the synthesis phase. And this is where you have that semi-conservative replication. And so the DNA divides itself from 1 to 2. Okay? And you replicate or copy your chromosomes. So think of your Xerox copier here. And if replication doesn't work, your cells won't divide. So if you don't get full replication, or if it only does half of it, the cell will not continue on. So it has to replicate completely in order for the cell to divide through mitosis. And we'll talk about that more in just a second. The second or third phase in this is called the G2 or gap 2 or growth 2 phase. Okay? This is where the cells continue to grow. They continue to grow until they're ready to divide into 2 cells, and that's mitosis. And when you get to the end of this G2 phase, your DNA starts to condense. And we're going to talk more about that next time when we look at mitosis and what actually happens to our DNA during this process. But at the end of the G2, we really jump into the mitotic phase, which is the division of cells. And we'll look at that, like I said, in a second, and then also further on in that lecture. The M phase, which I said is like between 11 and 12 o'clock, is where the cells actually go from 1 cell to 2. And next week, or the next lecture, we're actually going to look at the 4 distinct phases. We're going to spend some time looking at each one, what happens to the cell in each one of these phases. But this is just a nice little introduction to tell you that there are 4 distinct phases of mitosis, and we go through these things. And finally, when you reach the end of mitosis, your cytoplasm divides, and you finally get 2 new cells. Okay? So that's the typical cell cycle. 90% of it is interphase, where you go through the G1, the synthesis, and the G2. And then you have that little 10%, which is the mitosis, where your cells are ready to divide and become 2 new cells. All right? So that's the cell cycle. And during that cell cycle, if you look, this is just inverted the opposite way. Here's the G1, the S, and the G2. You can see we have these little stop signs. And what these little stop signs represent is that these are checkpoints. And if the cell hasn't completed the checkpoint, just like you have your checkoff sheet for the lab, it doesn't continue on. So after the G1, if the cell hasn't completed its task, it doesn't move on. It stays there until it can complete its task or it gets destroyed. Same thing with this G2 stop sign. If it doesn't complete its tasks, if they don't check off their tasks, they get destroyed. And a couple of those tasks include synthesis of their DNA. If they haven't replicated their DNA correctly, it doesn't go through. If they haven't grown to the right size, it doesn't go through. And if the cells just don't look right, they don't go through. Okay? And then finally, you have this last one during mitosis. This is called the M checkpoint. And basically that says if the cells aren't dividing correctly through mitosis, don't worry about it. We're getting rid of those cells. And so you don't want to have an abnormal number of chromosomes and everything else. And so if the cells just don't look right when they're dividing, they get eliminated. Okay? And what is this elimination called? Well, we call this apoptosis, and this is just a fancy word for meaning programmed cell death. And so when the cells divide, you undergo apoptosis. Okay, and it's basically a sequence of events in which the cells start to look abnormal, they kind of round up, balloon up, and they start blebbing off to the side. And you can see an apoptotic cell here compared to a normal cell. You see all these little blebs, it looks like a virus might have gotten it. And so what happens is little pieces or fragments come off the cell and eventually it just disintegrates into nothing. And that's typical. We have a lot of these things going on all the time because if the cell doesn't look right, we have these programs to eliminate it before it gets out of control. And so that's a good thing. All right? So what happens during apoptosis? Well, the cell is normal here, but if something doesn't look right, the cell will say, okay, it's time to destroy. And so what happens is the cell will get really rounded. So you see here it kind of looks jagged, kind of like what your fried egg cell would look like. But now you can kind of see it rounds itself up. It kind of looks like this big round ball, kind of gets inflated. Then the nucleus starts to separate out and you get these different pieces of nuclei throughout. And then the cell starts to bleb off and fragment. And then you get these little fragments that contain DNA and it starts to fall apart. And here you can see an apoptotic cell where you have all these little DNA fragments that are breaking off the cell. And that's how we normally get rid of these cells. This is a normal process that happens in our body all the time. And these cells destroy themselves and then we have our immune system that comes in and cleans up the damage. All right, so normal process. But what happens if this doesn't happen? Okay, so what happens if this doesn't work? Well, then we end up in a situation that we call cancer. And cancer is basically an abnormal growth of cells that just don't look right. And we call these tumors. Okay, these abnormal growths. And you can have two types. You can have these benign tumors that are not cancerous. These are just basically these encapsulated cells that grow abnormally and they grow bigger and bigger and bigger. And those are the things that your doctor will say, well, keep an eye on it. We'll keep an eye on it. We'll keep an eye on it. If it doesn't do anything to you, if it's not bothering you, not causing any pain, not causing any problems, they let them go until it does start to cause problems. And then they might remove it by cauterization or cutting it out or freezing it or doing something. The type that we worry about are these malignant. Malignant means that they just don't look right. These are ugly looking cells that just keep dividing out of control and there's no stop to them. They just don't look right, and that's the big thing. They invade other tissues, they break free. These are gross looking cells that you don't want in your body, and so that's what we worry about when we talk about cancer. So you either have benign tumors or malignant. The malignant ones are the ones you worry about. Okay? Typically, when you do get cancer, this is a gradual process. It's not like you're going to get cancer and die the next day. It tends to be a long period of time. And what happens is most people don't recognize the signs and symptoms or maybe not even have any signs and symptoms at that time and go and let it progress for a number of years before they do anything about it. And then that's when problems arise, is when you let it go for a long period of time. It's very rare that you'll ever have a cancer that will develop very quickly, rapidly, and almost kill someone. That getting a cancer and dying in 6 months is not real likely. Okay, doesn't normally happen. It does. I can't say that it never does because there are obviously incidents of when it does. But typically, it's a gradual process that most people don't even know until it reaches a stage and when they finally see some signs and symptoms involved with it. All right. And so what do cancer cells look like? And see, you can see here just by looking at this picture that cancer cells just look ugly. Here are your normal cells. They're nice and round. They have a beautiful nuclei. They differentiate into different things, but cancer cells are just ugly. They have these ugly looking nuclei, irregularly shaped. They may have multiple nuclei in the cell. They just don't look right. And that's basically what the doctor is looking for. These cells that have multiple nuclei, they look almost black, very ugly looking, don't look right. They don't have a definite shape to them. They're just not right. And that's what they're looking for in this case. So when they do a biopsy on the thing, they look for these really ugly cells. Okay? These cells don't undergo apoptosis. There's no stop to them until you remove these cells out of the body. All right? And so what are some of the characteristics? Well, cancer cells start with a normal cell, and something happens to them that they get mutated. They get exposed to something cancerous like UV or a chemical or a carcinogen, and then they start to grow abnormally. And you start to see these cells starting to grow into these tumor-likes. They continue to grow, and eventually, if they're not treated and taken care of, they can get into systems and spread. And when they spread that way, this is called metastasis. And metastasis means it's traveling, and that's a bad sign. When cancer starts to spread, then you're really dealing with some serious issues. And we'll talk more about that in just a second. Okay? So again, looking at the progression, you can see here, here's a normal cell that gets converted, some type of mutation to it. These cells start to grow a little bit out of control. You start to see these little tumor cells forming. And this is why they always tell you to check your skin and look at these things. Keep an eye on things. If something doesn't look right, get it checked out by the doctor. Because eventually what happens is these things start to grow and grow out of control. And when it reaches this stage, this becomes very serious, because if they start spreading through your lymph vessels or your blood vessels, this means now cancer cells can end up in other parts of your body, and this is really serious. You don't want cancer going to your liver, your intestines, other places, your brain, and other things like that. You want it to be localized so they can take care of it and get rid of it if you have a problem like this. Okay? And that's a bad thing. And so new cells can develop, travel through to different parts of your body, and that's how it spreads. And when it spreads, this is called metastasis. All right, and so you can look at the differences, and again, just comparing normal cells versus cancer cells. Normal cells just look normal. They have differentiation. They're normal. They undergo apoptosis. They avoid this, or they have contact inhibition. And all that means is when the cells are next to each other, they play nice. They don't start growing and pushing other cells out of the way. They just grow normally and happy, and if the cells are nice together, they don't grow out of control. In cancer, they don't care. They just start growing on top of each other, pushing each other out of the way, and this is when you get these ugly-looking tumors that start to grow in different spots, and so it loses that contact inhibition. They become multi-layered, disorganized, and then the biggest thing is that they can escape the area. When they escape, it's a bad thing that's called metastasis. So remember that. If things start to move, you're in trouble. So what causes cancer? Well, there's really two ways that cancer gets caused. You have these genes that you have inside your body that either promote cancer or discourage cancer. And most of the time, these genes that promote cancer, these oncogenes, are shut off. And until they get turned on by some mechanism, you don't have to worry about it. But if they do get turned on, that's when you have a problem. And we see this in cancer cells. These genes get turned on. The other type of genes actually suppress cancer cells. And again, these do a really good job of suppressing these from growing out of control and doing these bad things. However, if these genes get mutated, then they lose their ability to repress, and then you get this growing out of control stage. And that's what happens in these situations. So you have both progenes and repressor genes. So you have genes that get turned on and turned off. And if they do get turned on or turned off, that's when you get into trouble. Okay? Well, what causes these things to get turned on and turned off? Well, really, you have a number of different mechanisms. So you have these things called carcinogens. It could be from radiation, getting exposed to radiation and causing cancer. Could be from pesticides and herbicides, these chemicals that mutate DNA. Could be caused by viruses, and we'll talk more about viruses in Chapter 20. But what they do is they integrate their DNA into your DNA and can cause mutations to occur, and that can be a problem. One of those being HPV, and maybe you've heard of that before, where it causes HPV or cervical cancer and so that's another issue. And heredity. You might have the genes themselves. So a lot of you have heard of Angelina Jolie and that she had double mastectomy because she had these genes. And sometimes if you have a history of cancer in your family, you are now more at risk of getting cancer yourself. And so if you do have these genes, you want to be aware and keep a check on your body. So you might do things earlier than normal people would. Do mammograms and do blood testing and rectal testing and things like that to make sure that you don't have these cancer genes or cancer cells occurring without you knowing it. Okay? So to summarize, we have a cell cycle, and that has really two phases. You have the interphase and mitosis. The interphase is divided into three steps. The G1, which is the growth. The synthesis is where your DNA divides, or your DNA replicates, and then the G2, where the cell continues to grow. When the cell is ready to divide, you undergo what is called mitosis, and we're going to explain a lot more of that in the next lecture, so make sure you watch that one. You also have these checkpoints. If the cell doesn't look right, it says stop, and if it stops, it undergoes this thing called apoptosis, this programmed cell death. Happens all the time. You undergo apoptosis all the time because you don't want to have all these cancer cells going on that are uncontrolled. So you have a way to eliminate a lot of these cells through apoptosis. However, if this doesn't happen, if you don't have apoptosis and the checkpoints don't work, you develop these things called cancer. And again, basically what cancer is a mutated cell with uncontrolled growth. It means it's going to continue to grow and grow and grow. It's benign when it's non-cancerous, non-spreading, and it's malignant when it doesn't look right. And so again, there's no checkpoints, there's no apoptosis. These cells continue to grow, and there's no way to get rid of them unless you do some kind of treatment. So again, if you have any signs or symptoms of cancer, you want to eliminate it before it gets too late. Okay, and that's the rule of thumb. Always listen to your body. It's always important. Okay, and with that, if you have any questions, please feel free to ask me through an email. Send me an email or ask me questions in the lab, and I'll be happy to answer them about it. Make sure you catch the next lecture, which is on mitosis, and we'll go from there. If you have any questions, remember, just ask, and I'll see you next time. Thanks.